Biochemistry, Molecular Part 2. Functional organization of a eukaryotic gene. 1. DNA coding strand, transcription. 2. Pre-mRNA, splicing. 3. Mature mRNA, translation. 4. Protein. Regulation of gene expression, promoter. Site where RNA polymerase II and multiple other transcription factors bind to DNA upstream from gene locus. AT-rich upstream sequence, with TATA and CAT boxes, which differ between eukaryotes and prokaryotes. Promoters increase initiation of transcription. Promoter mutation commonly results in dramatic decrease in level of gene transcription. Enhancer. DNA locus where regulatory proteins, activators, bind, increasing expression of a gene on the same chromosome. Enhancers and silencers may be located close to, far from, or even within, in an intron, the gene whose expression they regulate. Silencer. DNA locus where regulatory proteins, repressors, bind, decreasing expression of a gene on the same chromosome. Epigenetics. Epigenetics is the changes made to gene expression without a change in underlying DNA sequence. Gene expression is heritable mitotically or meiotically. Primary mechanisms of epigenetic change include DNA methylation, histone modification, and non-coding RNA. RNA processing, eukaryotes. Initial transcript is called heterogeneous nuclear RNA. Heterogeneous nuclear RNA is then modified and becomes mRNA. The following processes occur in the nucleus: capping of 5' end, addition of 7-methylguanosine cap, co-transcriptional, polyadylation of 3' end, adding of poly-A tail, post-transcriptional, splicing out of introns, post-transcriptional. Capped, tailed, and spliced transcript is called mRNA. mRNA is transported out of nucleus to be translated in cytosol. mRNA quality control occurs at cytoplasmic processing bodies, P bodies, which contain exonucleases, decapping enzymes, and microRNAs. mRNAs may be degraded or stored in P bodies for future translation. Polyadylation signal. Mutation in polyadylation signal leads to early degradation prior to translation. Kozak sequence. Kozak sequence is the initiation site in most eukaryotic mRNA. It facilitates binding of small subunit of ribosome to mRNA. Mutations in Kozak sequence leads to impairment of initiation of translation, which leads to decreased protein synthesis. RNA polymerases. In eukaryotes, RNA polymerase I makes ribosomal RNA. It is the most common rampant type. Present only in nucleolus. RNA polymerase II makes messenger RNA, microRNA, and small nuclear RNA. RNA polymerase III makes 5-SR RNA, transfer RNA. RNA polymerases have no proofreading function, but can initiate chain synthesis. RNA polymerase II opens DNA at promoter site. RNA polymerase I, II, and III are numbered in the same order that their products are used in protein synthesis: ribosomal RNA, messenger RNA, then transfer RNA. Alpha-amanitin, found in Amanita phalloides, deathcap mushrooms, inhibits RNA polymerase II. Alpha-amanitin causes dysentery and severe hepatotoxicity if ingested. Dactynomycin inhibits RNA polymerase in both prokaryotes and eukaryotes. In prokaryotes, one RNA polymerase, multi-subunit complex, makes all three kinds of RNA. Rifamycins, rifampin, rifabutin, inhibit DNA-dependent RNA polymerase in prokaryotes. Introns versus exons. Exons contain the actual genetic information coding for protein or functional RNA. Introns do not code for protein but are important in regulation of gene expression. Different exons are frequently combined by alternative splicing to produce a larger number of unique proteins. Introns are intervening sequences and stay in the nucleus, whereas exons exit and are expressed. Alternative splicing. Alternative splicing can produce a variety of protein products from a single heterogeneous nuclear RNA sequence. For example, transmembrane versus secreted immunoglobulin, tropomyosin variants in muscle, dopamine receptors in the brain, and host defense evasion by tumor cells. Splicing of pre-RNA, part of process by which precursor mRNA is transformed into mature mRNA. Introns typically begin with GU and end with AG. Alterations in small nuclear ribonucleoprotein assembly can cause clinical disease. For example, in spinal muscular atrophy, small nuclear ribonucleoprotein assembly is affected due to decreased survival motor neuron protein, which leads to congenital degeneration of anterior horns of spinal cord, then symmetric weakness, hypotonia, or floppy baby syndrome. Small nuclear ribonucleoproteins are small nuclear RNAs bound to proteins, for example Smith, to form a spliceosome that cleaves pre-mRNA. Anti-U1 small nuclear ribonucleoprotein antibodies are associated with systemic lupus erythematosus, mixed connective tissue disease, and other rheumatic diseases. Primary transcript combines with small nuclear ribonucleoproteins and other proteins to form spliceosome. Cleavage at 5' splice site, lariat-shaped intermediate loop is generated. Cleavage at 3' splice site, lariat is released to precisely remove intron and join two exons. tRNA structure. tRNA has 75 to 90 nucleotides. Secondary structure, cloverleaf form. Anticodon end is opposite 3' amino acyl end. All tRNAs, both eukaryotic and prokaryotic, have CCA at 3' end, along with a high percentage of chemically modified bases. The amino acid can be bound to the 3' end of the tRNA. CCA can carry amino acids. tRNA contains the sequence necessary for tRNA ribosome binding: ribothymidine, pseudouridine, cytidine. tRNA tethers tRNA molecule to ribosome. dRNA contains dihydrouridine residues necessary for tRNA recognition by the correct aminoacyl tRNA synthetase. dRNA allows detection of the tRNA by aminoacyl tRNA synthetase. The attachment site, 3' ACC 5', is the amino acid acceptor site. Charging. Aminoacyl tRNA synthetase and binding of charged tRNA to the codon are responsible for the accuracy of amino acid selection. Aminoacyl tRNA synthetase uses ATP, one unique enzyme per respective amino acid. Aminoacyl RNA synthetase matches an amino acid to the tRNA by scrutinizing the amino acid before and after it binds to tRNA. If an incorrect amino acid is attached, the bond is hydrolyzed. A mischarged tRNA reads the usual codon but inserts the wrong amino acid. Start and stop codons. mRNA start codon. AUG inaugurates protein synthesis. In eukaryotes, AUG codes for methionine, which may be removed before translation is completed. In prokaryotes, AUG codes for N-formyl methionine. FMET stimulates neutrophil chemotaxis. mRNA stop codons are UGA, UAA, and UAG. They are recognized by release factors. You go away, you are away, you are gone. Protein synthesis. Initiation. 1. Eukaryotic initiation factors identify the 5' cap. 2. Eukaryotic initiation factors help assemble the 40S ribosomal subunit with the initiator tRNA. 3. The eukaryotic initiation factors released when the mRNA and the ribosomal 60S subunit assemble with the complex requires GTP. Eukaryotes, 40S plus 60S to 80S even. Prokaryotes, 30S plus 50S to 70S prime. Synthesis occurs from N-terminus to C-terminus. ATP, tRNA activation, charging. GTP, tRNA gripping and going places, translocation. Elongation. 1. Aminoacyl tRNA binds to A site, except for initiator methionine, which binds the P site. It requires an elongation factor and GTP. 2. Ribosomal RNA catalyzes peptide bond formation, transfers growing polypeptide to amino acid in A site. 3. Ribosome advances 3 nucleotides toward 3' end of mRNA, moving peptidyl tRNA to P site. Translocation. Think of going ape. A site, incoming aminoacyl RNA. P site, accommodates growing peptide. E site, holds empty tRNA as it exits. Elongation factors are targets of bacterial toxins, for example, diphtheria, Pseudomonas. Shine-Dalgarno sequence. Ribosomal binding site in prokaryotic mRNA. Recognized by 16S RNA in ribosomal subunit. Enables protein synthesis initiation by aligning ribosome with start codon so that code is read correctly. Termination. Eukaryotic release factors recognize the stop codon and halt translation. The completed polypeptide is released from the ribosome. Requires GTP. Posttranslational modifications. Trimming. Removal of N or C-terminal propeptides from zymogen to generate mature protein, for example, trypsinogen to trypsin. Covalent alterations. Phosphorylation, glycosylation, hydroxylation, methylation, acetylation, and ubiquitation. Chaperone protein. Intracellular protein involved in facilitating and maintaining protein folding. In yeast, heat shock proteins are constitutively expressed, but expression may increase with high temperatures, acidic pH, and hypoxia to prevent protein denaturing or misfolding.